So you're today with us with Sarah Farrant, president of the BCLA, and myself, Sònia Travé Huarte. I'm a researcher at Aston University, and today we'll be talking about photobiomodulation and saponification and one of the new clinical trials that we're doing. So we're going to take an approach where I'm going to be talking a bit about what we do, then Sarah about what she does, and then we're going to mix it all together. So we need to understand a bit about what microbiome is. So we all know about microbiome when we're talking about the guts, but microbiome can be found all over the body. We can have it on our skin, our lungs, in our mucosa. So what microbiome is a collection of microbes, bacteria, fungi that lives inside our body with their DNA as well. And it is inside us. So their functions are to protect us against pathogens, so anything that is coming to our body to react to it, to help our immune system develop and understand what is a trigger or not to the surface of our eye in this case. And also it helps us to produce and to digest that food that we're having. Now, the microbiome, the core one, is set on the first years in our life, but there's a lot of things that can change that microbiome, starting by aging, by diet, by medications, exposure that we have around, or anything that we put in or around our eyes. So what we need to understand that we don't understand just yet is how the microbiome in the whole body, not just our eyes, the gut, everywhere, how that reacts to environmental exposure, how those changes happen, and what can we do to understand what will happen or what will prevent or aid that change onto the microbiome. Now, looking into how microbiome affects the ocular surface, so we have a diversity of microbiome within the eye and even around the eyes in our lashes and lids on the skin. And there's so many things that can change what we have in the eye. We have the aging, obviously, as we age, we know that we have more chances of having Demodex. That also changes the flora around our lids and lashes. Environment, contact lens use, specifically hygiene. We need to be very careful in how we clean our lids and lashes. And the overall health as well, apart from medications that we've mentioned before. But they also have a protective role. So when we have something coming into our tear film or touching the eye, the first thing that microbiome will do is we'll get there, it'll try to understand what that is. Is it something that is going to compete against me? Is it something that I need to balance out? So sometimes there's competition even within the bacteria to understand, do I need to be here? Do I need to secrete something to inhibit certain substances? So that's the commensalism that we have. We have something of our eye that is normal to us, and then we have that relation with it. But what it does, it modulates homeostasis. So basically, it helps us understand with that immune response, it balances it out, and it prevents an excessive amount of inflammation. So it detects that inflammation, and then it acts upon it. Now, issues that we can have, dysbiosis, so when we have changes onto that flora that we have, that has an impact on the response on that onto the ocular surface. We can have, when we have blepharitis, when we have Demodex, when we have conjunctivitis, all of that is sort of like a cycle. One thing can lead to the other, so we need to be careful and manage these accordingly. So is microbiome good or bad? If we have an excess of bacteria, for example, when we have Demodex or bacterial growth, overgrowth, then we can have inflammation in the lid margin, and that will lead to having dry eye, having that inflammation, having more symptomatology. And even when the bodies of the Demodex itself, they die off or they remain onto the surface, that can cause extra toxicity on the skin. Now, do we want a complete eradication? Probably we don't want that either, because otherwise we would have no natural pathogen or like nothing that would act against those possible antigens coming. So we need to have a bit of the two of them. So we need to have a homeostasis of that microbiome. So now I'm going to pass this to Sarah, and she will carry on.
So when we consider our kind of normal ocular microbiome, it's quite interesting because bacteria typically outnumber us as our host by 10 to 1. But the ocular surface is quite unique. It's what we call paucibacterial, and what it means is basically it's a unique environment where we don't have nearly as many bacteria on our eye surface as we do in the rest of our body. So on the eye surface itself, we have 0.06 bacteria to one human cell. In the mouth, it's about 12 to 1. So our immune system has this really major role in maintaining this balance of resident bacteria. And the bacteria themselves also shape our immune system. So again, talking about that balance, they have a role to play with for us as well. So we have a host of sort of regular bacteria that make up our microbiome. And studies have kind of tried to align which bacteria are most commonly found on the eye surface. It depends on how they're cultured and how the bacteria are measured as to what we tend to find. And there are many different species, even on the eye surface, that we encounter in the research. So in dry eye disease, what the research is starting to find is that different types of dry eye disease will influence the levels of the ocular microbiome. So for example, in blepharitis, we know that things like staph and strep tend to increase. In MGD, we get decreases in staph. In dry eye disease, generally, we get increases in lots of things. So depending on what's going on and the roles of the dry eye disease itself, we notice changes in the flora and fauna on the surface. So I think it's time to really take stock about how we as clinicians are considering what blepharitis actually is. In practice, we see it, we see the collarettes, we see the build-up on the lid margins, and we see the knock-on effect on the ocular surface and dry eye disease that it contributes to. But we don't necessarily think about the role that bacteria in the microbiome are having on the lid margins. So the lids are, sadly in a way, a perfect home for bacteria. It's warm, it's moist, it's a nutrient-rich habitat. And blepharitis, whether it's frank or subtle, we encounter both types very commonly in practice, basically results in this overgrowth of the normal skin bacteria. What you get is this biofilm, and the biofilm increases the inflammatory markers, the cytokines, and triggers the classic inflammatory cascade that we see in dry eye disease. And the biofilm itself is this slimy film of bacteria, and it basically coats the community of bacteria itself to protect it. It's a kind of a defense mechanism, as it were, for the colony of bacteria that form. And once they get to a sort of certain size, a colony, they secrete this kind of sugary glycocalyx, and that basically gives them this protection, and they stick to each other, and then they start to influence each other's behavior, act more like a team rather than individual. And the biofilm also, unfortunately for us, acts as a protective armor. So it's actually harder to penetrate that once it gets to that certain level. The bacterial community, once they get going, they've got momentum, there's enough of a community there, they start to talk to each other. And it's called quorum sensing. And this is basically communication between that colony, and it causes gene activation. And in doing so, you get a release of virulence factors such as enzymes, lipase and proteases. And why does that matter? Well, we'll come on to that in a minute, but it's really important to consider. We also know that in clinic all the time, I'm sure Sònia would agree, we get a lot of cases where you'll look at an eye and they've got raging blepharitis, the bacterial load must be enormous, and yet the knock-on effect to their ocular surface is minimal. They don't have much inflammation, they don't have a lot going on. And why is that? Well, we actually believe that not all strains of things like Staphylococcus are identical. So some strains of staph, for example, are better designed to create a biofilm. So what happens is that matures rapidly. In doing so, it's releasing these inflammatory markers, but other strains are less virulent. So you're going to get variation on a theme in these different patients, which we see in practice all the time. Strains are more likely to be present in patients who get severe blepharitis and chalasia as a kid. So those kind of patients probably have strains of staph that are more likely to create biofilms. And other strains that produce slower growing biofilms, they're less virulent, less sort of cytokine inflammatory triggering. They might have those strains for their whole lifetime, and yet it never really gathers the same momentum. And so it really does depend on the strain that actually accumulates on a patient's lids as well. We know the bacteria need a food source, okay? And we also know that in meibomian gland dysfunction, the meibum that we secrete changes. So what happens is that the meibum goes from being unsaturated to saturated. And part of the problem with that is that the saturated fat that is resultant from the disease, the dysfunction, is a better food source for the bugs. Okay. We also know that the colonies that once they get to a certain size, they also secrete exotoxins as well as things like lipase. And exotoxins are designed by the colonies to kill off competition. Okay, so there's a bit of a war going on amongst various colonies. So they secrete these exotoxins, they're fighting against each other. Okay, the problem with these exotoxins is that they also cause powerful inflammation. So things like marginal keratitis is fundamentally triggered because these exotoxins are secreted. So the enzymes that bacteria secrete are designed to help them digest their food source. Okay, so what happens is it helps to break down the lipid to give them an easier meal, as it were. Okay, it's, I guess, bacteria's version of ultra processing, maybe. And it also helps them to penetrate their host better. Okay, so we know that Staphylococcus is really good at secreting lipase. And lipase breaks down lipids. So what happens on the surface of the eye, all this lipase is floating around, and it mixes with the lipid in the meibum, okay? And it's broken down. And the chemical process that we give that is called saponification, okay? Soap making in Latin. And because it is alkaline, that's the outcome is this process. And you can imagine, well, if we've got this self-perpetuating soap factory on our eyelids, that's actually going to cause a significant problem. The minute we mix grease and soap, we all know what happens when we're doing the washing up. It breaks it down. And that's exactly what we're trying to avoid when we've got dry eye disease. Okay, so you can consider it to be, unfortunately, yet another kind of vicious cycle in the programming. So when people have blepharitis, we know from studies that they tend to have about a 14 times increase in bacterial load. We know, as we mentioned earlier, that they're secreting these exotoxins to fight each other. We know that that inflames the host. We know also about the lipase, that breaks down the lipid, you're getting the saponification, that further breaks down the tears, that's adding to the inflammation. So this is a really important part of that, one of the elements of the vicious cycle of dry eye disease. So I wanted to run you through some videos just to help in the differential diagnosis. So you can see from this video, this is a close-up of the lower lid, that what you're seeing here is not saponification. I wanted to give you some differentials. What this is just an accumulation of bits of meibum, and the problem with MGD is the meibum tends not to mix very well, so you get it sort of accumulating in clumps along the lid margin. Then this is also not saponification. So what you have here, floating in the tear prism, what we call lipid bergs, and these are basically like little icebergs of lipid. And because the lipid composition is altered, it's now no longer mixing with the tears, and so you get these isolated clumps of lipid, which can be kind of confused with saponification. This is true saponification. So what you see now are bubbles, literally little foamy, frothy bubbles, and you can provoke them as well. So if you look at a lid margin, when you start to know what to look for, you can look at the lid margin before you express, and I'm a massive fan of expression, and you'll see not too much going on, nothing too obvious. The minute you start to squeeze and you're clearing out that saturated meibum, it mixes with the tears and you get much more saponification present. So I also now will grade saponification like at baseline as well as post-expression to see the differential in the clinical appearance. What we've actually devised as well now is a grading scale because one didn't exist. So we decided we needed one. So along with James Olson at Aston, we developed the notion of producing a grading scale to actually start to record this properly clinically in practice. And then I thought, well, is a grading scale enough? Well, probably not really. What I need as a clinician is a grading scale that tells me what to do. So what we've begun to develop, and it's still in research stages, so we're still understanding exactly how this will fit in, but we've developed a grading scale which also tries to give us a treatment protocol. Okay, so watch this space for how this will emerge. But you can see in the treatment protocol, when we have zero saponification, we're just carrying on with our normal, general management of dry eye disease, okay? But the minute we start to see that we've got a ocular microbiome dysbiosis, we're starting to consider how we can tackle that specific issue. So when it's mild, things like specific management of Demodex, hypochlorous, just simple strategies that are just starting to target the issues with that microbiome. When it gets potentially a bit more moderate, starting to get a little bit more aggressive with the options that we have. So considering IPL, considering shortwave blue low level light, which we'll talk about in a moment. And then obviously all the other options that we have on prescription. So watch this space for how this will develop over time, but we will start to publish some data once we've developed it on the scale. So the other element to the microbiome is obviously Demodex. So the problem with Demodex is that it is enormously common. By the time you are 70, you will have them living on you too, 100% prevalence. And as a direct consequence of Demodex being on your lid margin, there is an increase in bacterial load. They act as a vector for bacteria, so that bacteria live on them too, and then as they're moving around and migrating on your lid margin, they're moving around, shuffling in amongst that biofilm, okay? So we need to reduce them. And obviously things like terpinonforol or IPL definitely help reducing the general Demodex count as well. So when we consider low-level light itself consists of monochromatic waves, and they take different paths to lead to a common target. Basically, it's different to laser because laser has spatial and temporal synchronization, which low-level light doesn't. So shortwave blue specifically, what happens with my patients in practice, the moment I see any level of saponification, I started off a couple of years ago treating these patients with shortwave blue. Nowadays, I use shortwave blue on every patient I do low-level therapy with and light therapy because I see the difference that it makes to the bacterial load. So what happens with shortwave blue is that the actual blue light energy is absorbed by something called porphyrins in the bacteria. And that causes photodynamic inactivation. Basically, the membrane-bound porphyrins in the bacteria absorb the energy from the photon, and that disrupts them, breaks down the membrane and wipes out the bacteria, okay? And it's a weak defense mechanism that does nothing to human cells, but damages the bacterial membranes. So we have had some limited research so far, which helps us to understand that there is an impact on stabilizing that microbiome, okay, reducing that bacterial load. We know the bactericidal effect of light on different pathogens like Staphylococcus and leads to a reduction in that homeostatic imbalance, as it were. So there's a lot of exciting research that we can develop in this field to really look and engage at how we can utilize this for patients. So if we take this example here, a lady I saw in practice not so long ago, and she presented with a marginal keratitis. So immediately I'm thinking exotoxins, you just know pretty much straight away that there's a microbiome issue anyway. So we treated her with Maxidrol, it all got better, and then she came into my dry eye clinic. So when we started to properly look at the issues that she had quite a bit of Demodex Blepharitis, she had telangiectasia, she had gland dysfunction, as you can see on expression here, and she had, with me provoking nothing, just sat on the corner of her lid margin, quite a lot of saponification. She even had noticed it. She said, I get this kind of like little white bit of debris that builds up on my lid margin. And they do say that, quite a few patients do notice it. So in terms of her measurements, so we'd already had her on Maxitrol for a couple of weeks in that left eye with the keratitis. So her tear breakup time in that eye had already improved to about 17, 18. Her tear breakup in the right eye, which was so far untreated, was only 2. So you can see just in 2 weeks the difference that just putting her on some Maxitrol made. So clearly, a bit of antibiotic in there, a bit of steroid, and instantly we're getting a much more stable tear film. She had clearly some issues with her micrography scores, nothing too radical, but a bit of dropout there, and her tear meniscus height was fine. So the kind of working diagnosis, Demodex Blepharitis, MDD, She had a tendency for recurrent marginal keratitis. She'd had a couple before, and she also clearly had this microbial load issue, this saponification, this ocular dysbiosis. So we saw her in practice with a view to doing microblepharotic exfoliation, i.e. clear off the biofilm, just like plaque removal on your teeth. We just need to get rid of it. Okay, so we did that, and then we put her on IPL, which we did a sequence of 4 cumulative sessions, and then we also engaged in low-level light therapy, both near-infrared and shortwave blue. Okay, we then gave her some treatment for home use, so obviously warm compress, Amiga, some drops, and some Demodex wipes. So I then saw her 4 months after the conclusion of all of that, and you can see from the imaging, when I then did some expression, the meibomium coming out of her glands already in 4 months was transformed. It was much runnier, much more patent, and the lipid layer was much better as a result. So also what we noticed is that her saponification had literally gone. Okay, so there was no sign, and she had actually literally noticed it. I'm not getting the white stuff in the corners anymore. You know, it was that tangible. So then our plan moving forward was to repeat that process of treatment every 6 months. So my aim is always to hit a level where we're in a kind of sweet spot, and then kind of put it on cruise control, just keep them ticking over at that point, you know. And if they change, if something digresses, then just match that change what we're doing in the journey. And quite often what I'll find is that if things do change for a patient, then I'll just increase the frequency with which I'm seeing them and doing the light therapies and doing expression, and that just kind of pulls it back in again, and that can work really well. So this is just the image to show you just the difference between the pre-treatment levels of saponification and then 4 months later, just how interesting, how good it was looking. So Espansione have developed some protocols to help guide us as clinicians regarding how we should be using low-level light therapy in practice. And this is all available on the website as well. But in essence, what they're identifying is that the minute you have signs of blepharitis, signs of Demodex, or signs of saponification, you really want to be thinking about incorporating shortwave blue light into your low-level light treatment regime. But certainly I would go beyond that and say, well, nowadays I just make it part. I do a triad of light therapy. It's almost non-optional for me. If I'm going to manage them with light therapy, I want to be targeting that microbiome, and I just see it as a really kind of under-considered element of the management of dry eye that we need to start thinking a bit more holistically.
So we had more of the clinician side. Now I'm more of the researcher side, so I'll give you numbers because that's what we like. But before we get into that, I want to do a quick recap on inflammatory events, why inflammation happens, how it happens, and is it protected or is it not? So what is inflammation? Inflammation is a response of vascular tissues to something that is attacking them. It could be a tissue that is damaged, a tissue that is necrotic, but it's actually it's a protective response of the eye or of the body, and it's essential for survival. Now, the idea is for it to get rid of anything that has come into our system, to get rid of it, and then help with dealing with the consequences that they're left. How does it work? Well, the cells deliver proteins and on the specific tissues that they've been damaged, and then those cells are activated to then help heal the tissue that in turn is damaged. But without an inflammatory event, what would happen is that the infections would go unchecked. It would just pass the barrier straight away. The wounds would never heal because we don't have that barrier of protection. So they would constantly be then festering in there and being permanent sores. Now, the most important thing to me is to understand, when is it acute or when is it chronic? Now, an acute response or an innate immunity is when something happens, something's triggered, and then we have a quick reaction, very acute, painful. We have rubor, we have calor, we have the whole thing. But that is a solution. That's something that it's preventing us to go any further. So if we catch it on time, we solve it, it's fine. The issue then becomes when it becomes chronic, and it's always there. Maybe it's not as painful, but it will be always there. And just trying to deal with a chronic condition in that vicious circle is a lot more difficult. So the idea is for us to stabilize any inflammation whenever we can. And why we've started this trial? Well, inflammation, saponification, blepharitis, all of that has a huge issue with dry eye and any ocular surface disease. So if we look at this wheel from Badawin, what we have is that when there's blepharitis, we obviously have fluoride changes onto the lid margin, onto the lashes. And that in turn has an issue with the inflammation on the eyelid, apart from the whole eye. And then everything changes, lipid changes, tear film changes, stability of the tear film changes, everything becomes a bit of an issue. So we basically wanted to understand how to stabilize that microbiome on the lid margin, but also on the necks of the eye, on the skin, on the lashes. Because obviously if we have an overpopulation of either blepharitis or Demodex, we'll have a cascade of events which we don't want to get into. And we know that certain skin conditions can be more prone to having Demodex, to having blepharitis. So we need to treat the problem at source if we can. So we already have protocols on how to use photobiomodulation, but we wanted to understand things such as what's the difference between treatments. Is a red wavelength is going to give us more benefits just using red on MGD and dry eye? Or if we add a blue wavelength, how is the effect that we're going to have on the Demodex and blepharitis? And if we do, by how much will be the reduction of those colonies, of those bacteria? So that's where the numbers come in, and that's why we started the trial. As we've said, red light photobiomodulation is to stimulate the cellular function to keep those cells to produce a bit more. And the blue light is to help with the reduction of that bacteria, elimination or just reduction. So the other thing is that there's limited research on external tissues and how that is affected. So that's why we joined in. The idea of this clinical trial that we did is to compare red and blue, and then sham as well, to understand what's happening onto the microbiome, what's happening in terms of like blood vessels, what's happening around, as well as what the patients are feeling, symptomatology, and the signs that we observe when we look at lids, lashes, and ocular surface. So how is this study designed? It's a randomized control trial, which means that the main investigator was masked. He was not performing the treatments. So the main investigator was doing pre and post assessment. I myself was giving the treatments 3 times, and someone else was doing the lab work, so that we're all completely masked. There's complete separation. And it was a control one because we also used one of the arms was a sham. So I'll explain you that in a second. You can find the clinical trials in, this trial in clinicaltrials.gov. And if you have any patient that is willing to, we're more than welcome to have them. We're in Birmingham. The study is called REBEL, for red and blue light. And inclusion criteria, all of them must have dry eye, meibomian gland dysfunction, and blepharitis, either being by Demodex blepharitis or blepharitis on its own. And exclusion criteria, the normal exclusion criteria that we would have when you are to treat a patient for photobiomodulation. So how does it work? We have the first assessment, and if they pass the screening criteria, then they get randomized. After that, we do a swap that I'll explain you how we do it in a second, and then we perform the treatment. A week later, we do vision questionnaires again, just to see how the patient are feeling, and treatment. Same thing again. And then 1 month after that, we do the whole assessment again with the swaps. So at the first visit, on the treatment visit, we do the questionnaires just to understand how much the patients are feeling, pain, dryness, whatever they're feeling, so that we can have numbers as a baseline. And then we have 3 groups. The group A has 50 minutes of red photobiomodulation followed by 15 minutes. So it's red. Group B has red and blue. So those ones, one would expect microbiome to change more than probably do with the first one. Again, we don't know. And the third one is a sham, which means that there's still some light delivered, but the percentage is not as high as in any of those 2. So that is our control group, so we can compare it against. Clinical assessment on the first visit, we do questionnaires. I love questionnaires. I always do questionnaires. I need my numbers. And then we follow by tear film stability test, either volume or quality. So we have tear meniscus height and breakup time. I also look at the redness if we're looking at inflammation, even though it's not in the lid margin itself. We also look at the ocular surface. Lipid layer, because we know that the red could also activate those cells and the meibomian to flow out. Staining, lid margin, very important. We're looking at inflammation. We're trying to understand what's happening in that area. And meibomian glands, of course. So, apart from how the glands look, I want to see how those glands express. So we use a specific tool that gives a specific amount of pressure that's standardized every time, so that then we can grade how many of those glands are secreting and how they are secreting. So we have different grading scales for that. Again, I say I love numbers, I do. So, all the clinical assessments that we do, we look at the temperature of the lids just to see if there's any change during the treatments. We also do tear sampling, which will be analyzed in the future just to understand what is changing. Yeah, quite tricky. You need to be very steady in there. LACAM, so this device understands hemodynamics and blood, what's happening onto that lid margin. And saponification that Sarah just mentioned about before, we want to understand how much of these are we having, if we're having it at all, if it's changing. So we need to understand a bit more about it. Swabs. Another thing that we do is we assess really good the lid margin. Is it a colorate? Is it a Demodex? What is it? The tension that we apply for that. And then the swab. So in here we have a swab. And that swab, what we do, with the patient looking up, we just swab twice on the lid margin and along the lid. And then that's put onto a buffer solution. It's left in there from the time that we collect it until we go to the lab. And then we use that fluid to understand what's happening. But going back onto the Demodex, there's been a study that explains how to correctly look for it. So obviously, we're researchers, we need our science. So what we do is we remove that colorate that we have right in here, try and remove it, and then we move the lesion like an L traction. And that apparently, if there's the Demodex in there, it disturbs and then it pops out, so it's easier for us to see them. So that's one of the things that we do. Also, because we know that Demodex now is becoming a bit more dependent on us. So we need to understand what are we doing with them. We need to keep that microbiome in check. Now let's go into the lab work. So once we had that sample, we get the liquid from where the swab is and we place it onto an agar-agar plate. And that will put it into separate conditions and we'll let it grow to see if there's anything at all in there or maybe not. So 24 hours of growth. That's aerobic, which means that there's oxygen and it's at a body temperature, so 37 degrees, and that's left to growth for 24 hours. And then we also have the anaerobic, which is the same thing that you've seen before, but in this case, there's no oxygen. There's a huge bomb of oxygen here that pulls the oxygen out, so the growth of the bacteria might be different. And actually, indeed it is. So we have growth with oxygen and without oxygen. But one of the things that we want to check is, rather than how big the colonies are, how many there are in there. Because sometimes they might think, oh, well, maybe there's not as much. But if you count them, there's a lot of them. So we need to understand, are those numbers changing pre-treatment versus after treatment? And that's one of the things that we're aiming to do. Once we've grown those for 24 hours, what we do is we put some solution in there just to make it the medium. And then with the aerobic, we literally just scrape that bit from there, and same with the anaerobic. We get one of the colonies out, we rub it against that solution, and that's what helps us spread. So at least we have this controlled media. And then what we do is we tint it with different dyes. There's 3 dyes that we put, but it's to understand, is it gram-positive or gram-negative bacteria? Because they're very different. And as we said, Sarah mentioned it, with the blue light, they're aiming at specific type of gram bacteria, specifically one of them, because it's like more difficult. And then what we do is we evaporate everything. By this point, bacteria is dead. So we evaporate and then we pass this through the microscope. And we understand, is it gram-positive, is it gram-negative, and what is the impact of that? So we have patients that had the 2 types. The gram-negative tends to be more purplish, more pinkish, and the positive tends to be a bit more colorful, as you can see. But in that case, there's a mix. But what does that mean? So positive bacteria is more susceptible to antibiotics. But then when we have the negative bacteria, it's particularly concerning because it's a lot difficult, even with antibiotics, to help that. So basically, they resist a lot better certain treatments. So that's why we want to understand that, and we evaluate pre and post. Also, to understand, the gram-negative bacteria tend to have more risk of an infection in the eye, so we want to understand those changes. And then 4th visit, we repeat everything again. So expected outcomes, I'm just going to report 3 cases, but as of when I extracted the data, which was a month ago, we had 6 patients treated with red, 5 sham, and 5 that they were red and blue. So our hypothesis is that we're going to have less bacteria after the treatment, better clinical outcomes when we assess the ocular surface and the tear film, and that the patients also will feel less pain or less redness. So in here I have a couple of cases. This is one of the participants. So maybe it's not easy to see the difference in here, but the differences are happening in here. I highlighted them so they're easier to see. So what we have is that the first 2 questionnaires, there's a change from baseline to at the end of the treatment at 1 month. We also have changes in tear meniscus height, so tears. And we also have changes on conjunctival staining, corneal staining, and the frictional staining between the lid and the eye. If we look at MMP9, inflammation markers, in this case, it seems to go down, then it comes back up. We don't know, that's just one case. But also looking at aerobic bacteria, we have less than the one that we had to begin with. Second case, same thing is still happening. So I forgot to mention the first was red, and this is red-blue. So in this case, we still have benefits on what the patients are feeling. We have benefits on tears, and we still have benefits on all that it is corneal staining, so ocular damage. And we still have changes in bacterial count and MMP9. This one is sham, so it's red and blue, but both of them are sham. And we have some benefits, but we don't have as much as before. So conclusion of just these 3 reports, that doesn't mean that that's going to happen with all the study. We had more benefits, more improvements with the red and blue and red just on their own compared to the sham, which we were expecting, but the sham still delivers a tiny bit of light. So maybe we're doing something even with that percentage of light. So again, the conclusions in here are from those 3 cases only, but we need to do the whole 30 patients, and we're halfway through now. So what's the potential on the field of photobiomodulation? Well, first of all, we need to understand a bit more about the microbiome, what it is, and the function, and then understand if that is something that can help us as a diagnostic tool. And that can help us as well to use it as a tool against having that many issues in the surface of the eye. But if we look specifically at the ocular surface, there's been multiple reports and studies, as you can see in here, on photobiomodulation and these different diseases. So dry eye, meibomian gland dysfunction, wound healing, and ocular surface, and even keratopathy as well. So basically, photobiomodulation does have a potential treatment and promise as a therapeutic tool for those diseases. And now completely out of this, I myself had a study this morning. I had the red mask for 15 minutes, followed by the blue mask, 15 minutes, and it just like squeezed right out. So I don't want to say anything, but this is just my personal perspective, and it worked like a charm. So that is all for us. Thank you very much. And if you have any questions, we're here.
Shortwave blue low-level light therapy has a sort of unique set of strategies in which it works. So when we consider the role of dry eye disease and the kind of clinical importance that has with the dysbiosis of the ocular microbiome, we can start to think about utilizing shortwave blue therapy to actually start to help control that dysbiosis. When we have lost that homeostatic balance because of dry eye disease or because of anterior segment issues, that we can use shortwave blue to really help to regain that balance and reset the dysbiosis, to challenge the saponification and the bacterial load, to reduce the impact that the biofilm is having on the ocular surface and the consequential effect on inflammation that often has too. Aside from dry eye disease, low-level light therapy or photobiomodulation can treat a range of anterior ocular surface conditions. We also know it has a fantastic outcome for chalazions. We also know it's good for patients with rosacea as well as ocular rosacea. So there are a number of really exciting ways in which we can use the technology to help our patients.
So this trial that we're doing is a randomized control trial, which means that the investigator is masked to which treatment the patient's been given. And what we're looking at is what the patients are feeling. So the idea is for us to treat with different types of light, red, blue, and sham, which means a control arm. And then we assess all of those parameters before treatment. And then we'll see when we finish the 30 patients, we'll see what the outcomes are. So we had a couple of participants already that felt incredible benefits, starting for what they're feeling. So what they're feeling, which is that the symptomatology score has changed, even after right after the very last treatment. But even after 1 month of finishing that treatment, that patient still feels better. We also have certain cases where the aerobic bacteria has decreased, which is the bacteria that we have around our eyes when there's oxygen, which is our normal life. So that has decreased as well, and we will be looking into inflammation on our next stages.